discussion to exchange on the main theme of their workshop, which is computational challenges in very large-scale omics. And I would like to thank our 7 panelists for accepting to lead the discussion. So I'll give a very brief introduction to each of them. So we have Ana Conesa, who is a research professor at the Spanish National Research Council. Alejandra Medina Rivera, who is a principal investigator at the International Laboratory for Human Genome Research of the University Autónoma of Mexico. We have Katie Pollard, who is the director of the Gladstone Institute of Data Science and Biotechnology and also a professor at UCSF. Davide Risso, who is associate professor of statistical sciences at the University of Padova. Oliver Stegel, who is associate group leader of the German Cancer Research Center and also affiliated with EMBL and Heidelberg. Tandy Warno, professor and associate head of the Department of Computer Sciences at the University of Illinois, Urbana-Champaign. And then Jipin Wine, who is professor of biochemistry and molecular biotechnology at the University of Massachusetts. So I'd also like to thank all the participants for responding to our survey and for all their insightful comments on the questions that we asked. So before giving the mic to our panel and to just get the discussion going, I've tried to summarize some of the answers that we got to the survey. So one of the questions that we asked was, what are the most exciting questions in omics? And here are some of the answers that we got. One of them was definitely all the possibilities that the new technologies open up. So 3rd generation sequencing, spatial omics, single cell assays. And we heard already about that in the talks earlier today. Another exciting area of research is the integration of all these multimodal data sets to get a better understanding of biological systems, the integration of sample and population level data, and also the translation to the clinic. And we'll have more of that in a session later this week about the clinical and medical application. We also had a question about the challenges. And from the answer, it's clear that one of the challenges is to keep up with the technologies and the methods. There's a lot of rapid developments in the field. A lot of challenges related to data, in particular, the availability and the accessibility of data that are being generated. The quality of the data, we have missing data as well. The fact that there are very heterogeneous data formats and to some extent a lack of standards for storing and accessing the data, normalization and sparsity. There are also a lot of challenges related to how we analyze these data, in particular, how we assess the statistical properties of computational methods and algorithms, how we assess the quality of the results that we get from the data and the methods. So a lot to do in terms of benchmarking. A lot of challenges related to causal inference. So analyzing perturbation response data, defining outcomes, handling confounders. There were some comments also about how we needed to have a cultural shift in biomedical research where we think about modeling upstream rather than downstream of the study design and experiments. A lot of comments about computational reproducibility, dealing with scaling of the methods to handle these very large data sets, and also practical aspects related to the cost of storage and computing resources. We had a question also about how we can educate the next generation of scientists to be able to deal with all these challenges. One of the main themes in the answer was the importance of multidisciplinarity. And I think we have that already reflected in the audience and in the speakers for this workshop. In particular, there needs to be greater communication and collaboration between biologists, computer scientists, and statisticians. It's very important for the statisticians and computer scientists to learn about the domain, the questions that they're trying to address with the computational methods. Also to understand the limitation of the data, the biases and the noise. Exploratory data analysis, data cleaning are very important. We heard about that today with yours talk. There's a need for workflows that manage computational reproducibility, portability, and scalability. So we need to train our students to be able to develop these workflows. How can we do that? So a few people mentioned having summer schools like what we have today. And there were comments also about we need to empower students to be in the driver's seat in interdisciplinary teams. And then our last question on the survey was about the roles and relationships between academia, industry, and government. So in terms of academia, some of the roles that were mentioned was the development of new ideas and methods, basic research. For industry, roles that were mentioned were the generation of major applications and resources, translational aspects of research, standardization, and scaling. For governments, the roles that were mentioned was the regulation to ensure fairness and privacy, but not too much regulation. Funding for high-risk basic research, and then encouraging collaboration and data sharing. There were some comments to note that there were still very siloed areas of involvement of these three entities. They have different strengths and weaknesses. There's some successful examples of public-private partnership, but more needs to be done in that area. And the respondents noted that really, ideally, these three bodies should work together to advance science. So a few of the take-home messages in this last slide are there are clearly a lot of exciting questions and challenges in omics. It's hard to keep up with fast-paced development of new biotechnologies and methods. Data accessibility, quality, and integration are key. So we have data, we have methods, but how good are the results? Can we trust the results? We need to have collaboration, more collaboration with teams of experts from the relevant disciplines, and also greater collaboration between academia, industry, and government. So this is just a summary of the responses. And now I'd like to turn it over to our panelists and with the first question and see what they have to say about that. So what are the most exciting questions? Who would like to go first? Anna?
Thank you, Sandrine. Well, I have most exciting question. Okay, so but I guess that the exciting questions are also very much dependent on what is your field, right? So I think that I can probably talk about the exciting questions on the things that I do in the field that I work more. These days, I'm working a lot on third-generation sequencing technologies for transcriptome analysis. And I think this is creating exciting possibilities for research. One of the things that I am seeing here is that these methodologies, for me, they are changing the way I'm looking at gene expression and then transcriptomes, because they are offering you a kind of mind-blowing view of transcriptomes as a very dynamic process, and we have been talking a little bit of there have been talks about that too, and I think that they are going to be challenging the way in which we define what is a transcriptome. We are used to think of reference annotations, of expressed genes and transcript that have a defined structure. And then I think that these technologies will be indicating that it's really a very dynamic process. And then we have to learn how to annotate this dynamic process and create the reference annotations, how we are going to be doing that in the future. I think that's going to be a challenge. When we are seeing experiments on these technologies, they are generating and they are claiming that they are having, I don't know, like tens of thousands of new transcripts being expressed in human. Are we going to be really annotating all this? I think that's going to be one of the questions that we have to address in the future. But probably also is the same, you know, for single cell data. So how we annotate the data that we obtain from these experiments. So I think that's a question, right? Because the huge amount of diversity that we are seeing with every single experiment, this is something that we want to report. So what do we have to keep and what do we have to discard? That will be kind of a question. And in connecting with that, for me, also a very important question is how we translate all these models, and we have seen a lot of modeling here today, into actual understanding of the underlying biology. And that's true for multimodal data, but in general for data. I think that we are very good in creating networks and creating distributions and of goods. I think that we are doing this a lot, but if you think of from all the today's sessions, so what were the biological insights that were clear? Actually, we have been talking very little about that, right? So maybe there was, one exon here that was somehow related to autism, but that's not really bringing us much closer to understanding biological mechanisms and biological processes. So I think that we have to do much more in that area because we have data, a lot of data, we have models, but understanding of the biology, I think that this is where we are still failing. And in connection with that, I think that there has to be, so we really need to have much more the experts in biology being part of the data intensive research. And we have been talking about about education and so forth. I think it's great to have multidisciplinary teams. I think that's great and we need that. But I also think that especially biologists need to have education that is data intensive and that the data intensive field becomes part of the education program, especially in biology, because we cannot do biology anymore if it's not data intensive, right? So the collaboration is great, but I think that we need to have that component there. And I'm going to be stopping now and letting other people talk.
Thank you very much for all these points on and Katie will address.
Yeah, I just wanted to jump in on the point about mechanisms briefly, and then we should all take turns talking. I think that was a really great point. I work mostly in the area of genetics and how changes in DNA cause changes in phenotype, like what makes a human different from a chimpanzee, or what does somebody get sick and someone else doesn't. And we've had tools for a while to, at a population level, if variants are common enough and effect sizes are big enough, to sort of associate genetic changes with phenotypes, but not to really understand the mechanistic steps through which that happens. And I feel like understanding the mechanism is the key if we actually want to use that information for good. And so very excited right now. I think we are at a time that just was not possible until recently to be able to go from genome to sort of phenotypes of the genome itself, like epigenetic features, 3D genome folding. They're great models now that predict those things very accurately from single nucleotide changes in DNA. From there to things like gene expression or other cellular phenotypes, from cellular phenotypes to tissues and tissues to organisms. I think that a lot of the field does one of those little pieces, and the exciting thing for the next 10 years is to actually put all of those things together. So like Gian and I both work a lot on that first step of DNA, these deep learning models, and we can you can give me any genetic variant and I can tell you what it'll do kind of thing, right? But then how do we go from that to like the cellular phenotype, cells to tissues, tissues to organisms in a mechanistic way, I think is very exciting. And that's a fun open challenge for all of us that to try to think about how to solve very rigorously. Go ahead, Tandy, you're up.
I'm going to follow up on Katie very briefly. So I'm really interested in similar ideas, but pushing deep evolution. So being able to do deep evolution means you have to be able to do things where the evolutionary processes are much more complex. We have a lot of models for doing this. We have a lot of software for doing this, but the models haven't been reinterpreted in the aim of going to deep evolution, and they haven't been really made more realistic. And so to be able to really go at the tree of life, which isn't a tree, but a network, but to be able to do this, we need much more biology informed modeling. And we need to take advantage of these assembled genomes. So there is a potential for real breakthroughs in how organisms evolve, how they co-evolve, how they adapt to their environments in detailed ways that we can't do now. But we need to do much more about the modeling and then the inference methods. So I'm very excited about the possibilities.
Thanks, Sandy.
Could I add from what Katie said earlier? So I think we're at a point that we can answer something that's really, I think, really important, which is the differential question. So if you look at the cell, if you look across the transcriptome, it's reasonably easy to predict the different expression of genes within a transcriptome. But I think the differential between two different kinds of transcriptome, maybe between patients and controls, between two perturbations, that's much, much harder to predict. Like, it's easier to maybe predict the differential from variants to open chromatin, maybe 3D chromatin structure. But by the time we go to differential expression, like among the projects we have looked at, the predictability of differential expression is really bad. So somehow these
Going from
sequence to expression.
Precisely. But
Yeah.
we could do the middle step. And
Exactly.
Have a hierarchical model.
Exactly. So
It
I
makes
think,
more sense.
yeah, that is with the data we have right now across single cell, more causative modeling, we're at a step to answer this question, which I think is going to make a big difference in our understanding.
Yeah, I mean, just to quickly add to this, I think what's also really nice, seeing over just today is, how we can piece the different elements together, right? I mean, from, sequence to accessibility to transcription. The other one is also regulatory networks, how genes dependencies on, how they're interlinked, and how we can bring those components into bringing this. And I think what in the end should be really an interesting goal is to also look at how we can use the data to understand how tissues function, and going, the next step, I think going to an organism is maybe, at least a complex organism, a big step, but at least at a sort of tissue level communication, I think there's a lot to be done by combining regulatory networks, variation to accessibility to transcription, and causal discovery. Spatial omics, which I think we'll hear about more later, will be very important to understand how cells interact in context towards causation. And what maybe could think about as a community as well, how we could gear our modeling and approaches that these individual problems we're solving, that they are designed in such a pluggable way that we can piece them together to be bigger models. I'm not talking about simulating a whole cell type of approaches that I think very ambitious, but I think we see more towards that, that we can piece individual modeling components that solve sub-problems. We still heard a lot about very specific sub-problems today, how we can piece those together to solve bigger problems. And that's also something to think about, how to foster this interoperability of these components, that this is possible, common languages, do we use probabilistic programming frameworks? I think that's an important question to be able to do what you all alluded to, which I completely agree with. Thanks.
Yeah, so I completely agree. And I think that we have to rethink a little bit what we do when we do like data integration, right? Right now, a lot of these data integration things are like just looking for shared latent variables, right? But like, to me, the real power of putting together all these technologies is like, for example, I have a high C data and I can predict TADs, and then I look for an answers only within the TAD because I know that they're kind of like enriched there. So like trying to little bit like what the other panelists have said, like putting back the sort of biological question at the beginning, right? And then trying to use these as tools to answer a real biological question rather than just we have a bunch of data and we want to kind of like see what the data tell us, right? The other thing that Oliver mentioned briefly is like spatial transcriptomics, right? And so going back to the original question, to me, that's very, very exciting new technology that will be very transformative in the next few years, especially like, for example, Lear in his talk had this Murphish data. And I think that those like Murphish type of technologies are really great because they can give you like even like subcellular sort of like transcript coordinates of the the different transcripts and how they interact, and then like the bigger question of like how cells interact to each other, and that to me, it's also kind of transformative in the sense of how we deal with data. Like these are huge images that are going to require like terabytes of space, and like we want to probably like quickly being able to have like a bird's eye view to look at like how cell interact, and then zoom in a cell and see how a transcript interact. That I don't think we have like the bioinformatic knowledge to be able to do that in an efficient way yet. So I think that that'll be interesting to see how the field moves into that direction.
Just quickly, Anna already mentioned this a bit, but one of the challenges also will be how do we agglomerate all these data sets that are being generated? And then the issue won't be even the fact that they were done by different labs or different machines or technologies. It would be that they are not even annotated in any similar way. So metadata is one of the major challenges now in these endeavors, because there's data that's useless because you have 100 libraries and you don't even know who's male, female, or patient on control because they just submitted a table saying 50 and 50, and that's useless. So how us as a community can push our collaborators or ourselves towards annotating data in a more consistent way so we can someday use it all together and create really complete maps and atlases.
Thank you, Alejandra. Anyone else from the panel would like to comment? I think we've already jumped to challenges already. So both the exciting problems and the challenges are clearly associated. So we had a lot of insight from the panel. Does anybody?
Yeah, maybe I can still add. Maybe we have enough data to understand some diseases, because I think if you compare patients versus controls, very often a lot of things are different, but I doubt all of them matter. So if you want to have some kind of treatment, if you could find out what's going on and what really matters, I think that could be an interesting challenge, so that we can just target therapeutics toward the difference that matters.
Yeah, I wanted to bring up a challenge that's sort of a cultural challenge in science, which is that all of you very smart people, I bet like 99% of the time are looking at data after an experiment has already been done. Maybe you helped design the experiment a little bit, but we're either mining public databases, a collaborator comes and asks for help, or something along these lines. And so imagine instead that all the modeling people were had some great models that linked, sequences to epigenetic features and to phenotypes, all the things we've just been talking about. And our models could generate hypotheses, and those hypotheses is what was driving data collection. Now, that sounds like not that crazy, but it actually is kind of crazy because nobody wants to do that. Like, it's just, there's a huge cultural resistance to it. And so the challenge I see is for us to develop some examples where that works really well, and we get a lot more value out of the data that was generated than if we had just been playing a more passive or downstream role. As soon as we have a couple of really big examples, and I'm saying this because I'm very confident that having modelers driving the process would make a huge difference. If we can do that and show that we predict something awesome, like you were just describing, Ziping, people will pay attention.
Maybe just to comment on that, I think this is very achievable. I mean, on the one hand, because the culture is slowly changing, computational becomes more data-driven, but also technology. I mean, soon, there are now technologies, high throughput perturbation screen, other elements where it's just not longer feasible to make ad hoc choices about what perturbations to consider. Modeling-based design criteria for experiments will be more and more important. So I think we have to work collectively to be in the loop, to really put the computation, ultimately even objective algorithmic decision functions into the decision point of this. And I think also when selecting projects where this is a goal, I think are maybe also more future directed, something we could focus on. So this is, I think, a really, really achievable vision, and I completely agree. That this is something.
Putting a positive spin on it. I love that.
Thanks, Laura. Anand, Sandeep.
So I think, well, I just wanted to add one additional component. So we have been seeing here some talks relating to to measurements of in large, of more or less large patient cohorts, and then we are looking at the genomes and of the single cells. There are initiatives all over the place of sequencing more and more individuals, with the hope that we can then find the variants that are associated to diseases. And I think this is generating a huge amount of data that will be very valuable. But I think that we are really missing a very important part of the equation, which is the measurement of the environment of these individuals. So we know that phenotypes is genome plus environment, right? So, and in many of these of diseases that are high prevalence diseases, there is a very strong environmental component that is on top of the genetic variation. And I know that we have been, and people working on putting together all the health data and all the information that is available for these patients that you are sequencing. But actually, there is a lot of this environmental impact that we are not measuring. That is, the lifestyle of the person, the life history. So where have you been? What have you done? What have you eaten? How you have exercised? Which air have you breathed in? Everything that is around you, that actually is having a huge impact of what's happening to you, right? And I think that now we are entering an era in which, there is a lot of monitoring systems around us. And I think that we also need to know how to leverage this data to integrate it with the molecular information to really come into predictive models of the phenotype. And I think if we don't really think of how systematically measure the environment, I mean, we can sequence million of individuals, but I don't know if we will get there.
Yeah. Oh, sorry.
Okay, so I'll be quick. I actually sort of said this earlier, but the challenge that I want to bring up is related to what I think is also the potential. So in my own area, which is phylogeny estimation, people do phylogeny estimation using standard models that have been around for decades. And the assumption is that it's good enough. The assumption is that it's just a question about NP hardness and coming up with effective heuristics, and it's fine. I actually don't think it's fine. And I think that we really need to have some concern about those estimation problems where we don't really know what the right biological model is. And other than simulations, we don't have benchmarks. And the simulations are problematic because you simulate under the model and you test under the model. That doesn't tell you much. So I really think we need to get some concern. I think it would be great if people were worried, because then we might actually test it. So earlier I saw, I think it was you showed a simulation where you simulate under a complex model, but a simple model is fine. Great. That's not being done in my area. Okay. Even the complex models are simple. And the realism in biology is so much more heterogeneous than any of the models. So I really get worried about this. And it's even worse for multiple sequence alignment. And that is worse for genomes. So I think we just need to be worried. Instead of assuming we have software, as long as we keep getting the same results, it's fine.
Thanks very much.
Yeah, so I wanted.
Some terrific points being made on that.
Yeah, no, I know. It's I wanted to react to what Anna was saying, but then now I also have a point for Tanya. But I think they sort of like they're similar, which is sort of like jumping a lot ahead to the question of like research, sort of like academia and in industry, and they can sort of collaborate. And I think, like, for example, a very good example of what you were saying is like what 23andMe has been doing, right? 23andMe has been like collecting these genotypes of a lot of people or a lot of customers, right? And then they keep sending them questionnaires, right? And a lot of people answer to those, right? And so they have a much better idea of their cohort of, you know, what they do. And if they come up with a new question, to just send out the new questionnaire of their email. So like, that's a good model that maybe we could borrow in academia. And, you know, going back again to this kind of like academia versus industry, like I spent maybe like 50% of my time, 1 year of my postdoc with a startup company here in the Bay Area, right? And like this company had a FDA approved genetic tests for a certain type of cancer, right? And the amount of like quality of the data and the modeling they had to do in order to pass this sort of FDA approval is something that you don't often see in academia, right? Like in academia, it's very often to see like this sort of double dipping problem. You just run a lot of models on your data until, you know, you get the one that has the highest F1 score or the RSC or whatever. You know, if you want to get actual like FDA approval for something, like you have to like just get part of the data, just never use it again, like, and just, you can do all the training you want on your training set, but then the moment you start using your test set, then you're done. You cannot touch the model anymore, right? So people are much more careful in like avoiding these sort of like overfitting problems. So, you know, these are kind of a couple of examples of things that maybe, you know, we could learn as, you know, it would be nice if there were papers done like that in academia as well, right? Which kind of maybe goes a little bit beyond just simulations and showing that simulations work.
Thanks, Stephanie.
Yeah, maybe a related point to what David mentioned was starting one step earlier is this sort of accessibility and standards for using common data to solve problems. And there, I mean, you know, Leo in his talk highlighted this resource, you know, creating this, and I think there are other community efforts. And I think investing in this area could be something, it's maybe also an interaction with industry where there could be common interests to having these resource standards established and using them and having data, you know, uniformly processed, available for everyone, and solving some of the tedious elements. I mean, from a European perspective, there are these 4 magic letters, GDPR, which causing us a lot of headache for human data, and regulation is slightly easier on this side of the Atlantic, but you know, respecting data privacy, but still making data accessible for small labs with overcoming huge regulatory hurdles, which companies can obviously afford by spending money. And I think that'd be very important to solve solutions that are of mutual interest to avoid a lockout that academia can't do certain things because the hurdles, the expense is just too high to get that implemented. I think that's a real challenge where one could ever think about creative solutions with commercial entities.
So I think this is a great discussion, and what I wanted to mention was that I think it's important that we are not mixing up basic research and translational research and translation activities too much. Because, I mean, in this omics field, many of the data types and data sets, they have been generated just to see what they actually could be used for, not because there was a hypothesis that you knew upfront that you would be able to deal with. When we switch over to the translational aims, I mean, there is a whole regulatory system that actually is trying to control what you actually expose to the patients. And if we have decision support systems, we need to get them approved as medical devices, both in Europe and here. And I think that's a good thing because it's very dangerous to let algorithms run stuff without having it replicated in different countries and find out how good these models are to generalize. So I think we should discriminate a little bit what is blue sky basic research and what are the translation names. And then I'm sure there are many surprises that suddenly we can see that something could reach the translational stage much faster than we actually had anticipated, but it's still two different things. Then I also wanted to mention one other thing, and that is that I've heard that, I mean, almost in all industries or activities in society that involve technology, whenever a technology has reached perfection, it's phased out. I mean, when Terry Speed was finished cleaning up the statistical analysis of spotted arrays, they went away. And then Affymetrix came in, and then it's changed it was a game changer because now there was a new type of data with less noise and boom, boom, boom. And so I think we should get used to it instead of just expecting that all the sort of new developments that they can be standardized up front, because I'm not sure it's possible. But these are my two comments: basic research, translation, but also that newer approaches also.
Thanks very much for these 2 points, Søren. And I think Davide has something.
I wanted just
add.
to briefly comment on the second point, which I think it's very, very good. And I think we also, though, shouldn't, just throw away everything that we've been learning from these older technologies, right? Because like it's a little bit like what Lior showed in the second part of, or I guess the third part of your talk, where, there's, for example, like moving one technology further, like bulk RNA-seq, we learned a lot about, negative binomial models and like, these other sort of quantitative biology fields that are still pretty much valid in single cell analysis, and they will be valid for spatial transcriptomics and for long read sequencing. So it's, I remember Rafael Izzari once said that, you guys that do single cell RNA-seq, then, you just need to read my stuff on like quantile normalization of RNA-seq and of microarrays, and you'll be fine, right? That's all like a special case of that. But it's a little bit like that, right? We should don't forget kind of the older literature, especially for methods. I think that's important.
Reinvent the wheel.
Exactly. Yeah.
I think this reinventing the wheel is really a important challenge. Also, conceptualizing the problems in such a level that they can be applied to lots of settings. I think this count matrix processing problem is a good example, as you mentioned. It's around for a while and probably will be around for some time. But then there are also sub-problems which are more technologically specific. And I personally find it as a challenge really hard to scout out for technology. Which one is going to be the one? When do you jump in developing this book method? Which problems solve themselves? We discussed now long read error rates in nanopore, and there's a question: do we develop a model, or do we just wait until the next chemistry comes out and the error rate drops by an order of magnitude? And I think these sort of decision functions are real challenges, and it's also difficult to decide for individual scientists in the community, where the interface between technology overriding computational solutions, because they're no longer a problem, deconvolution of spatial transcriptomics, when spots are subcellular resolution, maybe you don't want to deconvolve anymore. I mean, all these decision functions are not so trivial. And there will be solutions that have a longer or shorter longevity, and sometimes it's not so easy to predict how long the problem will be around until a disruptive advance just wipes it away. So I think that is actually remains a challenge. And this generalizable concept is certainly a good way to make sure your science is future-proof, so to say.
Yeah, just to push my other point about models to drive experimental design or collection of data, I think models should be driving technology development. We shouldn't be waiting to see what the next thing is that can be measured. Where are we performing badly? Where are the blind spots? What piece of data do we not have? And can we try to push for that to actually be collected in the future?
Well, I think that for most computational biologists has been the other way around. So the technology came, and then we have to to see what can we do with that, right? So the technology goes ahead, and I agree that. I mean, we have to pose problems to technology, but the reality is that it's kind of the other way around. But yeah, I think that. It's hard to say. I mean, if you as a computational biology of a data analyst start addressing the error model of the data and spend time with that, and then the technology advances, and then what you are doing is worthless, right? So I think this is something that you have to live with, because that's gonna be there all the time, and then you have to be fast, or the technology will catch up for sure, right? Or the technology will put another problem, that's for sure. Well, I'm trying to see which are the things that are kind of recurrent through the technology advances, right? And yes, we are measuring molecules, and we are measuring more and more molecules, and more different type of molecules, and with different technologies. But I think there are some things that we are still having as challenges that we have not been able to address properly in the last 20 years, right? And one, again, I insist, is biological interpretation of the data, models, right? So regardless whether your original data was microarrays or if it was single cell, it's still very hard to create a mechanistic model of what you are seeing, of the biological system that you are seeing, right? So I think that we are not progressing fast enough there, right? And that's regardless of the technology, how you measure these. And probably it's because it's hard for the human brain to, to understand all these molecules working together, right? So we need to come up with more creative ways of understanding the data, right? And I was going to be saying something else, but I forgot. So maybe we'll come up to this later. But again, so I think that there is the technology, and the technology goes ahead, right? And then there are some things that we are kind of we don't get there. And one of these are the interpretation. I think is the great challenge that we have in front of us.
Yeah. So I just wanted to add on a couple of points that have been mentioned sometimes now. So one is like, which data sets should we address now? No, like, and this tempting idea of thinking that now we have a lot of data sets, so we maybe don't need much more to understand patients and controls. But actually, yeah, maybe that's true if you are only focusing in North European ancestry populations, then maybe that's true. But if you want to learn anything else about like the other part of the world, then maybe that's completely false, because we also don't even have data on lifestyles, diets that come from any other place in the world. Like once I asked someone from the UK Biobank, how would they address diversity? And they told me, we have migrants. I'm like, yeah, migrants have a particular way of life, like they change environments. But what of the other people that remain home? Those are lost in life, and probably will never know what happens in the disease for that population and in that environment. And so at some point that is tempting, and I think at some point it will also bring information on how the disease interacts with the environments, with the changing environments. If you characterize different populations for the same disease, you could learn more biological insights. There's even medicines that are developed in the States that promise, for example, remission for lupus in 6 months, that we see at least anecdotal, that they are not working in the Mexican population. We have no idea if it's just the patient we are surveying, or if it's just that it's actually, there is a difference in the kind of lupus or in the environment or the genetics, we don't know. So there's still a lot of data that has to be collected across the globe. And sometimes it's tempting to think that our methods and the things we develop work enough in this data and these models. But maybe we still need to address other data sets. And
Thank you, Alejandra. So I think one more comment.
One last comment.
One other comment. I just want to make sure we get to the next few
Yeah,
questions.
of course.
We're
still in question
which
one.
is great. And I think the message I get from all of you is that it's not just small challenges, but a lot of them require paradigm shifts. So that will lead us to the next question, which is how we train the next generation for that, and then get back to the discussion about government, industry, and academia. And then finally, Alejandra and others have alluded to that, how we make sure that the research and education is accessible to populations that are typically underrepresented in science. So I'm just making sure I repeat these three important questions so that you can address them in whichever order you want, because I really like the dynamics on the panel and all the things that are mentioned. So.
Okay.
Yeah.
I just want to add something really brief
Yes, of course,
about
yeah.
making our effort and the research future-proof, right? I think that's really important. There's nothing worse than spending time chasing after technology. So I think we should define our research problems broadly enough, and then always revisit it when new technologies become available. Very often we could answer a piece of the problem, but when new technology becomes available, we should say, are we answering more than before? Are we adding anything rather than reinventing the whole thing again? Right. So with single cell, are we answering additional questions before when we had a bulk sequencing? Now we have single sequencing. Are we answering actually biological questions at a higher level? So I really like the idea of being future proof.
Well, maybe I can get started on education. So, I mean, I guess I'll give my somewhat niche, maybe experience, in the sense that I'm in a stats department in Italy, and I think, I'm the only one in this room. But like, no, I mean, more seriously, I guess, it is obvious, I think, to all of us that interdisciplinarity is the key, right? So I've been hearing from other panelists that, biologists need to be trained to work with data. But, if you want to be a good, like, sort of bioinformatician or applied biostatistician, it's the other way around too. Like, you have to be trained in like biology and understanding biological concepts. And that's, to me, for example, that's a struggle for my students as, someone in the stats department, which is not in Berkeley, which is, obviously much more, advanced in that sense. You have the Center for Computational Biologists. There's a lot of crosstalk between the stats department and other departments. It's not like that in every university. And even, when I was applying for faculty jobs in a lot of places, they would just, tell me I didn't have a biometrica or, a stats journal publication. And that's still true for my students as well, right? So I think there needs to be somewhat a shift towards accepting sort of like multidisciplinarity as a way to kind of promote students and promote young faculty, and like not as a, I feel like in my career, I oftentimes had to work twice as much to prove that I was like, a worthy statistician, or in addition to being like a bioinformatician. And I think if you change statistician to computer scientist or to biology, it's still true, right? I mean, if you're a computational biology, maybe you have to prove that you're still like a true biologist. If you're a computer scientist working in bioinformatics, you still have to kind of like show that you go to computer science conferences. So I think that's the mind shift that we need to make sure it happens. I don't know how, but like, I think, maybe it's a good discussion point.
I just want to completely agree with you, and just want to say one of the things that's really great in statistics is you guys actually look at data. In computer science, it's rare. So I think that really getting computer scientists to work on data early and to really think about it and to interpret data is incredibly informative to their development. But you're right, unless the academic departments can start to value people who don't look core like them, it's going to be hard. So I think that's particularly hard in math departments. It's not as bad in CS, except some CS departments are still way behind. It will be interesting to see what happens in the future, but I think we need to keep pushing on this, and the academy needs to change.
So I have something more on training, but I don't know if someone wants to follow up on.
Okay, sorry. So my main worry is like, it's going to take students longer and longer training until the student becomes productive. So if that period goes to 5 years, we're all done for, right? So how do we solve this problem? How do we bring like education as quickly as possible so that we can start to be productive? So I think that is a challenge in our field.
Yeah, I mean, related to this comment, also what Davide mentioned, I think it's interesting how to define interdisciplinarity. I mean, it's interdisciplinarity that you work between two currently classical university departments, or do you create new fields? And there are different approaches. So, creating new programs, computational biology programs that sort of live on their own, have their own identity, and it's not so clear to me what's the right approach. When is a new field? When do you spark these fields, and how do they evolve it? Or do you stick to some basic disciplines and foster the interfaces between those? And it looks like in our area, there's probably everything. We have these different bridging between different classical domains, stats to biology, and then there's new programs like computational biology have their own identity. And I guess one needs to foster a tolerance of these different career paths. Students being able to switch is very important. I have huge headaches getting my students registered if they come from the wrong background, have the wrong whatever. Then getting them, enrolled to these programs, and these things, these hurdles, they just cause frustration. So I'm really with Davide that we need to figure out how to fix it. On the other hand, it's also important that somehow there is a community that respects what you do. Otherwise, it becomes sort of the accountability is also an issue. If nobody understands what you're doing, then you're sort of lost in between. That's also hard to fix intrinsically.
I think also that between the multidisciplinarity, so at some point, we cannot be experts on anything, though we do have sometimes to prove ourselves in many fields to gain some respect. But there is a point where you have to stop and acknowledge that for some biological questions, you have a biologist or someone that is really an expert on that area, that enough to interpret the data and to communicate, which is very important. But you won't be the expert in that area. And I'm really in between all the time. And I think I know enough computer sciences to understand the methods and to use them, but I wouldn't be able to create a new one or modify like the statistics behind them. So I collaborate with people that are good at that. And I acknowledge that I won't be doing that.
Thank you, Alejandra. So Jessica?
Thank you very much for the discussion. I just have 3 points to make regarding this training. I think first of all, from today's talks, I think many of us agree that benchmarking is much needed for our field, but I feel like probably for our students' career development, benchmarking work is undervalued, right? If a job candidate has benchmark papers, many people or departments may treat these as less important than novel method development. That's something we need to address as a field. And the second point I want to make is regarding the interdisciplinary thing, because I'm in the statistics department, but I have to say my department is very open-minded and I feel very fortunate about that. But one way I found to interact with my colleagues who are not in this field is to talk about the importance of our work or like the impact of our work, because I think that's something everybody can appreciate. And I think biomedical sciences are particularly strong for our impacts, right? Nobody will argue that treating diseases is not important. And I think the third point is about how do we, like in our field, I can also see we have people who focus more on algorithms, method development, and people who focus more on biology like answering questions using computation analysis. I think we need appreciation within our field, because I've seen people who think developing algorithms may be better, intellectually better, but I think we need to appreciate the importance of having new biological questions. On the other hand, I think for people who focus more on biology, they also need to appreciate the need for new algorithms. We wouldn't think that everything can be solved by existing algorithms. So I think within our field, this mutual understanding and appreciation is very needed.
Thank you very much.
I just want to follow up on that very good point. There's a big problem with how do you evaluate researchers? There's too much emphasis on citations. And if you focus on citations, you're going to think anyone who gets a lot of biology papers is much more important than, let's say, a theoretical computer scientist. You can't even normalize for fields. So it's very tricky. And there's something has to be done so that understanding contributions is made with greater intelligence. Also in science papers, the computer scientists, the statisticians, the mathematicians are never first authors. So there's this other issue. So I think this is a complex issue. It's sociological, and it's not going to be answered very easily.
I just wanted to jump in real quick. I'm delighted you mentioned that point, Tanti, about assessing contribution. We're making progress, I think, gradually. I mean, it's better than 20 years ago when I was in the job market and I felt like I had to give 2 job talks, a theorem proof talk and then the applied talk, which had much more impact. So, but it's still a big issue in statistics. I was at the joint statistical meetings last summer, and there was a whole session devoted to elevating applied statistics. So young scientists, young statisticians are still struggling with that issue. And I think assessing it, the contribution, applied journals, collaborative project and so on. So we need to be better than that. And we're facing this on a very large scale beyond biology here at Berkeley and other campuses with the creation of data science department. So that's a really big topic, and I'm delighted to hear that being addressed on the panel. So I thought I'd just jump in with that.
Yeah, you already mentioned the key points actually. Yeah, but I think there's also some things in our own hands, and when we write author contribution sections and we think about papers, to reflect that, be really careful about how we attribute credit, and also how to guide trainees and how they write their CVs. That you really have a couple of papers where you clearly state what was your contribution. Some grant agencies now use this also, write down 5 papers with contribution sections. And I think there are ways to do that, but we have to think also sort of as a community do the sort of bottom-up approach. While obviously there needs to be top structures need to be fixed. And what's clear from this is also, particularly if science is so collaborative now, that this linear author orders is just sort of such a thing of the past. Sometimes it really doesn't make any sense anymore. And I really like all these papers where people, write down author order determined by coin flip or other things to just make the point that this is just insane. And I think that's a mindset we should all try to sort of propagate and communicate. Yeah.
Oh no, I was just raising my hand. I've done the coin flip.
Okay. Okay. Anna.
Well, maybe we need a benchmark on how to report the contributions, right? Of the author. No, and I'm saying that because in the last consortium project in which I was involved, we kind of made a very strong effort in monitoring the contribution of everybody and having kind of groups of authors that have similar. So we were kind of applying almost a clustering algorithm to group the authors based on the contributions that were in a huge Excel sheet in which we have a contribution and authors, and then you have to put a cross in which kind of contribution as an author you have. So, and I thought that was actually a very interesting, exercise, right? And maybe we need to have also papers discussing of which is a good way to describe that, so that people can adjust to some kind of fair guidelines of how to report contributions.
But yeah, no, I agree. But I wanted to go back to I guess Oliver's question about like whether we need a new discipline or not. And I think in some sense, having like a bioinformatics discipline would be like recognized at the same sort of level as, you know, statistics and biology, and like that would make our life easier in some sense, right? The fact that, you know, we as a community, like we could set up our own rules on like, you know, hiring people and like giving promotion and all that. And of course, it's also dependent on the places, right? For example, I was in a biostats department at Weill Cornell, and it was clear from day one that they, you know, as biostatisticians, we were not expected to be like, you know, PI on R1 or like last name of papers to be promoted to associate, right? Because they recognize that biostatisticians have a different role. But it's not like that in every school, right? And yeah, so I don't know.
So there's also a question on so these new disciplines, and that's something that has been happening in the last 20 years. It's the creation of like bachelor level new degrees, like on bioinformatics. There's some already, but from the bachelor level, and that goes into what you were saying, Shipping, about when do you start training them so they can be productive someday. Yeah. So yeah, exactly. So these bachelors, well, those aim to really train people in bioinformatics, so they know the biology and they know the computational sciences, but they are not expert in neither. That's my end point. But they can start working and being like in between the two areas, and then do graduate school and maybe specialize a little bit more into one side. So maybe that's a solution to start like creating these fields from the bachelor levels, and then they will exist as institutes and that are more inclusive with people that really understand from the beginning how this area works, how this career works, like which kind of papers do you get, how do you, well, yeah, the authorship can be discussed and it should be benchmarked. That sounds good. But yeah, if there is a field that it's being created, maybe it can be already within our programs of education.
Yeah, I mean, I just want to elaborate on what I said earlier about training. I feel like students come in, you know, the data sets are so big and so much pre-processing, normalization this way, that way, and then data quality. I mean, just so much to learn, which is unprescribed. How do we educate students so that they can figure it out as quickly as possible and get to the point where they can contribute rather than mechanically, but intellectually, right? I mean, I feel like I struggle with my own students. Some of them, I have to kind of carve out a project and plug them in and say, okay, just work on this part, okay? Just don't worry about the big picture, because if I don't do that, that student is not going to graduate. But how do we struggle with this kind of training? I think these are very real issues.
I just wanted to give an example. Okay, I just wanted to give an example of actually at the University of Florida, when I have been a professor for 7 years. And so at some point, the founder of NVIDIA, who was a graduate student from the University of Florida, came to the university and put money on the table to hire 500 faculty in artificial intelligence. 500. So what is happening there now is that there is this movement across the university in which every single discipline will have someone in artificial intelligence around. And that's biology, that's architecture, that's even the library, you know. And the idea of the movement that is going on is that actually all these quantitative skills should be part of the curriculum of every single class. So we are not talking about adding new classes so that every student will have to learn, I don't know, Bayesian statistics. No, we are not talking about that. We are talking that if you are working in your lab, maybe your lab journal is something that you do in R. I don't know, just to give a very simple example. So that the quantitative skills are transversals and the quantitative quantitative way of thinking is transversal to every single class, right? And this is being now implemented like with a lot of $1 million on the system. But I think that probably we have to think that in the future, and I think that we are facing a future in which data is just an integral part of our lives, right? So, and that's what we are seeing. It's all about data. So that will be part of the curriculum in a very more transversal ways, not as a, as a particular class that you take, but something that is embedded in even a single class. And I think that's how we have to train the future students.
Yeah, and I guess going back to what Alejandra was saying before, like, I'm a statistician. I will always be a statistician. I will never have the biology knowledge of a biologist, and kind of like the same for biologists with statistics. I think what we need is sort of like giving enough to have a common knowledge and be able to collaborate and talk the same language when they, when the students for your group and my group get together to write a paper, right?
Yeah, I've really enjoyed this discussion, just reflecting on what you have said, Katie, especially, but others also. It's made me think a little bit about the relationship between biology and physics. So I feel like in physics, there are well-specified and really precise models that have very specific predictions, and that allows people to design experiments and technology to test those predictions. And I just wonder if panelists could comment on, I know it is kind of aspirational to imagine developing like really models that are more in line with physics, but I feel like if we could, it could lead us towards some of the goals that you all have described.
So I had been leading a large bioinformatics center for many years, but then 10, 15 years ago, I got the offer to make a large omics effort together with a colleague, especially in proteomics. And but this should be at a medical school. I used to be at a technical university. And then I started to do MD, PhD students. And to turn it around. And it's a little bit what David also said. Instead of us teaching the students to become multidisciplinary, we could turn it around and then they could teach the teachers how to become knowledgeable about things in a different area. And it's been a very interesting experience because medicine is super reductionistic for a good reason. If you have one disease, you want to focus on that and what can you do for this patient? We come with our omics mindset. We want to look at all genes and all tissues, all cell types and so on. But a lot of medicine wants to focus. And so that has been a sort of an eye-opener for me that these MD, PhD students, they can really change your mind about things. And then it's also interesting that they run away from you again because they don't want to become computational biologists. They want to go back into the clinic and become surgeons and many other things. But then they have this upgrade in terms of data handling and mining patient records and so on. So so I think, like David said, we should maybe get the PIs a little bit out of the equation. And I really liked what you had on the slide, that you should let young people lead the multidisciplinary effort, because I think at least I've tried that without knowing that was what it was called, but I think it was a good idea. Yeah.
I won't take credit for it.
No, I wanted to say I have exactly the same experience. So every time that I have been working with a medical doctor, and the funny thing is that they come to you first because they have all this omics data they are so excited about, right? And then when you bring a kind of complex model to them, they are scared, and then they come back and say, yeah, but which is the biomarker, right? So because it's kind of too complex, right? So one way they want like both, right? So they want that you look at everything, but at the end of the day that you come with a simple question, with a simple answer, right? So that's the paradox a little bit, right? And I have failed always that I have when I have provided my multi-omics of omics analysis results to a medical doctor and ask for help in the interpretation of the data, they were never able to give me that, right? Because it was too complex. So I think that this is a two-way thing, right? So we need to understand from them what they need, but I think also that at some point there is a need for some more quantitative training of these people that are looking into complex diseases and want to see, more. So you need that this training is multidisciplinarity in the conversation, but a little bit more trying to reach the knowledge domain of the other to be able to communicate better and to be able to give back what they are asking you that you give them.
Yeah, so I really like the idea of this transversal learning. Like at some point, everyone should touch a bit of data analysis or like doing research. And so when I was doing the postdoc in Canada in CKIDS, I saw the MD PhDs and how they interact with the clinical part and the research part, and it was amazing. I go back to Mexico, and honestly, the medical community is a bit more worried about people not dying. So it's really hard to make them collaborate with you in research because they also haven't done research seriously ever. They had like a little class maybe in the undergrad. In Mexico, MD school is undergrad level. So maybe they had like one class of scientific methods and that was it. And so it's a struggle because you want them to collaborate so we can do like very good level genomic science with patients in Mexico, but it's also hard to make them collaborate because they don't see the point. Because they like reading the papers, but they don't really understand why should that be done in Mexico. So like that's a challenge, but having transversal classes would make a difference.
So I wanted to transition from this topic to the next topic, which has to do with how do we do our training so that we actually engage people from other groups, from underrepresented groups, et cetera. I want to say that even though you want people to understand the biology, you want them to understand the statistics, you want them to be good at the computer science, it's too much to ask. And what I think is perhaps a very good thing to think about is that many of us have entered into this field gradually. We've entered into it from where we're comfortable. So I started as a graph algorithms person. Ron, you were probably the same, right? So there are a bunch of us who have entered into this field and gradually gotten hooked and gradually made our ways into being able to be contributing things that are valuable. If we have the same patience, we can actually do great things. And that would include taking people who are smart and driven, but not knowledgeable about biology, statistics, and computer science, and giving them opportunities to get involved in projects and to participate and to gradually get hooked and to eventually make really big contributions. But I don't think we should be thinking about they have to start as undergraduates and then we have to be able to do this and that. We'll never make a difference in terms of diversity in our communities if we have such a standard.
Thanks, Fundy. And I think Ron.
Yeah, one historical perspective that may be helpful. Physics became aware of mathematics about around 1900. And it took, I don't know, 50 years. And by now, there is a applied mathematics department, which are actually doing the kind of physics, the mathematics of physics. And practically every physicist is an applied mathematician. But there is this difference. So the more advanced, the more method development is done in applied math department. But on the other hand, every physicist is mathematics aware. And I think this will happen, is happening gradually to biology as well, that more and more biologists are becoming aware of computation, while at the same time, we still need the professionals in departments of computational biology. It's a long process, but I think we are heading this way.
Yeah, I think it was going back to the previous comments about interdisciplinarity and having people to do that. And I just wanted to second, these interactions that I think if you want to foster these changes quickly, then using the next generation and enabling them to do that is a very good way. I mean, we've been at the MBL had this interdisciplinary postdoc programs between two laboratories. And I think the best experience of people in the lab, if they come in and do this science that I would never do and the other lab would never do, and then they do it between these two labs and field and bring things forward. It's been super enriching. And it's excellent for the fellow because they're enabled, they have a lot more control and ownership of their science. And it's super exciting for the participating labs because they get pushed in new directions. So I think these tandem relationships between fields, between labs and enabling trainees to break these bridges is maybe a way to save a little bit of time, although I agree it will take time to get there.
Comments from the panel?
Just had a comment about the increasing diversity and the students, and I thought it was a really interesting point. And one thing I've seen, I worked with a lot of students actually who came from Mexico, from UNAM, which has a really great undergraduate program in computational biology, really phenomenal. And one thing I've appreciated is that if you can get the students, productive in a research area quicker, then there are labs who are happy to have them and pay for them. And I think that that's one consideration that we have to think about for these students, which is, they can't do a master's that they're going to pay for while they retrain in a different area and then spend a little bit more time. And, the family sometimes expects that, they're going to support themselves and they have expectations. And so if we can get them to a point that labs will appreciate them quicker, then they can have more runway to do exactly what you were saying, which is to, broaden slowly their expertise base, which is a long process, but we kind of need to get them there just because of the practicalities of, the way, funding works and their own economic situation. So something I've observed.
Yeah, it's interesting. I've thought a lot about this over my career, and to some of Xiping's points, you can't train somebody in everything. It just becomes impossible. And I think the best strength that I've seen, or the sort of the most success I've seen, is when you have the right group of experts, each with their own depth, working together and speaking the same language. But that's not someone walks into the office, we solve a problem, and a month later it's over. These are like 5-year, 10-year collaborations that are, at least in my experience, the ones that finally reach fruition. And so, and Tanya, I really love what you said. I think the story that I've been hearing again and again from talking to people about how do we increase diversity in genomics, for example, is it's not around teaching someone a class or a couple classes, but it's about creating pathways that they can follow. And that means not just, creating the educational opportunities for them to take and to follow their own path. And some might become deep experts, some might be multidisciplinary, but also creating training environments where they see people that look like them, teaching the classes, creating research opportunities, right? It's very much about the experience. What I'm hearing again and again from my colleagues, it's really more about, after a student takes a class, that it's clear for them what the next step is, the next step is, the next step is, and giving them that path and making that path easier for people to follow allows them to engage in these 5, 10, 15-year efforts that it takes to really maximize.
So that is a very good point. And actually, it's something that I've been discussing with some peers in Latin America, because there's a lot of funding for these capacity building courses. There's a lot. We appreciate it. Don't stop doing them, please. But at some point, these capacity building hardly translate into collaborations. It's like people will come, they jump, they teach a class for a week, they train them in genomics, bioinformatics, NGS, whatever. And then you see grants from these people where they say, I have been teaching in Uruguay for 1,000 years and they are awesome. And it's like, okay, but you have no collaborations from that country. So what have you been doing there? You go teach, you never talk to them again. So these paths, like how do we keep training these people and investing in that capacity building? And actually at some point, bring them into being collaborators. These are smart people. They just happen to be in a lower advantage position, but how can we bring them into being real collaborators and not just receivers of our knowledge? But those paths, I would like them to be created. I don't know how to, but at least that's a discussion that I think we should be having.
Yeah, so maybe some kind of long-term community, like people will buy in and precisely like this program we're in now. People come every several years and renew friendship, making new friends, and then it needs to have a sustained life.
One of the things that the Italian government does for their PhD scholarships that they offer is that during your PhD you're allowed up to 18 months in a lab abroad, like in a group abroad, right? That's actually how I went to work with Sandrine, and I ended up here, right? And that to me is, really a good model, because it goes a little bit of what Oliver said before, where, if two labs collaborate, then you're creating a new science that maybe the individual labs didn't think of. And, these two labs, if they're like from, one side, maybe a less advantaged country, and then they come to the US and the Western Europe, then you can really build these sort of like collaborations that then maybe they go back, they graduate, and when they graduate, they can do a postdoc. Or, they at least have like, papers or other collaborations to show that they have like done good science with, so that to me would be like, if there's US funding to kind of like, that would be like something like a semester or a full year visiting programs. And it's free, right? I mean, the thing is like, my advisor at the time just emailed Sandrine without knowing her. And it's like, do you want a free student? She said, yes, right? Because, I mean, why not?
One like huge specific problem is that you can't get NIH fellowships or NSF fellowships if you're not an American citizen. So that means half the students in my lab are totally ineligible. We can probably collectively pressure politicians to try to change that. I think it's the kind of thing that makes perfect sense to change. And if we could get that accomplished in 5 years, it would, I think, be transformative for bringing more diverse audiences into at least the universities in America.
That's a really good point, in particular training grants.
Those are really would be terrific tools, but they're only permanent residents or US citizens. So that would be a good thing to change. Yeah.
We have
5 minutes left. The panel, the session went by so fast. So I don't know if anyone has concluding comments.
I appreciate the comment about a sustained relationship. I think a lot of what people were talking about was about the pipeline and getting people in or checking boxes. But the key is to have people stay in the fields and do great work, and for the environment to be supportive. There's no point to have a pipeline if people feel like they don't belong.
Exactly.
We have a lot of work to do there.
Yeah, I think at the end, human capital is the best capital.
Wow, that was a lot of terrific comments, very lively, insightful discussion. So really thank you so much to all of you for contributing all your thoughts to the discussion, and thanks to all the participants in the audience. I really enjoyed the panel, a lot of things to think about. So big round of applause for our panelists. So that's it for the first day. So our next